Fatality rate at 18.6%. Severe fever with thrombocytopenia syndrome is an emerging infectious disease caused by severe fever with thrombocytopenia syndrome virus. The SFTSV is transmitted through tick bites, saliva, and blood from tick-bitten animals or patients. No vaccines or treatments for SFTS are currently available, and only symptomatic therapy is offered. Since diagnostic technology required in this field has not even been developed, in some worst cases, patients don't get proper intervention when it is necessary, or they spread the virus to others. A rapid SFTS diagnosis technique enabled by the CRISPR-Cas system. Developed by a research team led by Professor Yoon-jae Song at the Gachon University, this highly available and reliable technology will write a new chapter of the innovative diagnosis and prevention of SFTS. This technology utilizes Cas12, one of the CRISPR-Cas proteins known for genome editing. Cas12a CRISPR-RNA complex can recognize target DNA using complementary CRISPR-RNA to the target DNA sequence. After recognition, Cas12a is activated and induces non-specific single-strand DNA cleavage. The heating unextracted diagnostic samples to obliterate nucleases process is used in genome extraction. When a sample is placed in an extraction buffer containing TCEP and EDTA, then heated, the nucleases present in the sample are inactivated. Also, the viral particles are lysed and the viral genome is extracted. An RT-RPA amplification method in isothermal conditions is used for gene amplification. The process of synthesizing cDNA from RNA and amplifying the cDNA is carried out quickly inside of a single tube. LB-Cas12a, used for genome detection, is a CRISPR-Cas protein derived from Lactobacillus bacteria. When an LB-Cas12a CRISPR-RNA complex combined with CRISPR-RNA, which was created by referring to the specific base sequence of the SFTS virus, binds to complementary DNA, a collateral transcleavage activity is activated and randomly cleaves surrounding single-stranded DNA. Here, if fluorescently treated single-strand DNA is added, the presence of the virus can be detected with a change in the fluorescence value when the ssDNA is cut. The biggest advantage of this technology is accuracy and convenience. LB-Cas12a detects only genomes with photospacer-adjacent motif sequences. The two CRISPR-RNA-RPA primer sets, S1 and S2, developed by the research team, achieved very high testing accuracy by using base sequences that are commonly found in all 6 genotypes of the FSTS virus, but not found in other viruses or bacteria. This technology uses an RT-RPA amplification method instead of RT-PCR, which is time-consuming and expensive. RT-RPA is capable of isothermal amplification, so testing can be performed on-site with simple equipment without transporting the specimen. In addition to the fluorescence detection method, the research team also developed a lateral flow assay to detect the SFTS virus. Designed specifically for SFTSV diagnostics, the LFA kit maximizes the convenience of testing. This technology was recognized for its academic value with its reference in an SCI-level journal published in 2022. We have recognized its value in the industry since we have completed the domestic patent application and PCT registration processes and are currently at the international patent application phase.
Severe fever with the thrombocytopenia syndrome virus known as SFTS-V infection, is increasing on an annual basis globally and is becoming a serious public health burden. To prevent the spread of SFTS-V infection, we need a rapid diagnosis and systematic quarantine. The problem is that SFTS-V cases occur in rural and remote areas with difficult access to healthcare. Thus, a simple and inexpensive field-deployable diagnostic test kit for SFTS-V infection is demanded. We designed and developed a rapid and sensitive diagnostic method using a CRISPR-Cas 12A system, and this method can be applied in fields such as rural healthcare center or military-based infirmary without using inexpensive equipment like a thermocycler.
A rapid SFTS diagnosis technique enabled by the CRISPR-Cas system. This technology will ensure rapid and accurate diagnosis of SFTS and in turn will contribute to treating our patients and preventing the spread of SFTS-V.